Hello and welcome to Science Research Weekly, Season 4, Episode 35, Little Red Dots and Exo-Neptune Ridges. Get ready for maize shoot stem cells, a 5-image Einstein cross to identify dark matter halos, identification of little red dots, ATREIDES for exo-Neptunes, Volume 114 of the Journal of Statistical Software, ggplot 2 geoms and stats, and classic scientific paper review. This episode covers materials from September 12th to September 18th. Let's get started with some research articles. First up, large-scale single-cell profiling of stem cells identifies redundant regulators of shoot development and yield, trait variation. When you think of stem cells, you probably think of humans or animals, but plants have stem cells too, especially in their shoots. But it's hard to capture them, observe them, study them. Here, researchers were able to optimize a protocol to recover thousands of maize shoots cells, advancing the study of shoot stem cells. Next up, HER-S3, an exceptional Einstein cross, reveals a massive dark matter halo. So an Einstein cross is an astronomical optical feature where light from a distant galaxy is bent by gravity from the object in front of it, sort of bending it around, so that creates 4 images. But astronomers found one that actually had 5 images, the 5th image being in the center, and fear that it actually must be due to a massive dark matter halo in the way. Staying in space, the next article is a remarkable ruby absorption in dense gas rather than evolved stars drives the extreme Balmer break of a little red dot at Z3.5. There's a strange type of object in cosmology. It's compact, it's red, and it's high, and it has a high redshift source. It's called a little red dot. Wasn't quite sure what they are. If they were stars, they're really, really dense stars, and that didn't really make a lot of sense. So it turns out that they actually might be a theoretical object known as a black hole star, where the dense gas surrounding a black hole is accreted and releases energy. And that's what it kind of goes through here. And continuing our space trek, ATREIDES 1, embarking on a trek across the exo-Neptunian landscape with the TOI 421 system. So researchers look at the distribution of exo-Neptunes, so those are planets outside our solar system around the Neptune size, so that's 20x size of Earth or more. And they really were distinguishing regions around a star for exo-Neptune abundance. So really close in is what they call the desert, where there's none. Whereas farther out, there's a savanna where there's some exo-Neptunes, but then between the two, there's this area that they call the ridge, a region they call the ridge, where there's an overabundance of exo-Neptunes. And so figuring this out kind of helps shake out how planetary development works, how solar systems are typically orchestrated. All right, with that, let's turn to our research tools. Volume 114 of the Journal of Statistical Software is now available with a variety of articles like Nubo, a transparent Python package for Bayesian optimization, mDendro, an R package for extended agglomerative hierarchical clustering, EBNM, an R package for solving the empirical Bayes normal means problem using a variety of prior families, exploring data subsets with B-tree, HMMTMD, hidden Markov models with flexible covariate effects in R, high Bayes, an R package to fit individual level, summary level, and single-step Bayesian regression models for genomic prediction and genome-wide association studies. Further along, there's TSCI, two-stage curvature identification for a causal inference with invalid instrument in R. There's quantile regression under limited dependent variables and stata. GLM-CAT, an R package for generalized linear models for categorical responses. There's optimum allocation for adaptive multi-wave sampling in R, the R package Optimal. And finally, mixed effects additive transformation models with the R package TRAM-ME. Three new CRAN packages, three new R bloggers posts for today. First up, from R OpenSci, all the ways to programmatically edit or parse R Markdown / Quarto documents. So there's things like cold chunking, linear programming, template tools for boilerplate documents, string manipulation tools, abstract representations, etc. Second, from R on Every Day is a School Day, yet another layman exploration of bioinformatics called Learning About Assembling DNA, Blast, and Loci Sequence Testing, MLST, a note to myself. So I really have been enjoying this series. And then finally, from Stats What It's All About, exploring ggplot2's geoms and stats. So a good refresher, good orientation to how those two play nice and work together. All right, 4 new CRAN packages that might be of interest. There is Henna, a versatile visualization suite. CCAR3, canonical correlation analysis via reduced rank regression. Serve GME, analysis of survival data under graphical and measurement error models. And Workspace, managed collections of datasets and objects. So there are no new grants of note. However, given the free space you have not chasing down grants, check out my new bi-monthly series, Classic Scientific Paper Review on YouTube, where I review classic scientific papers. The first one up is 6 red flags for suspect work. Little red dots, great big like button. Thanks for joining me for Science Research Weekly. Science on.